various kind of cells, eukaryotic cells and prokaryotic cells. Now next we shall go for the next topic, that are various organelles. Again, you have studied it since long. You have studied it in your earlier classes before joining pharmacy course, and also you have studied in different subjects as we have discussed. Let us go for these classifications, or what are these organelles. So it was actually the term was coined long back, almost 2 centuries ago, that since human body or any animal body is made up of several organs, and all these organs have different functions. Similarly, cell has got, and cell is a very small unit. So instead of calling it organs of a cell, because cell itself is a part of organs, so instead of calling it as a organs of cell, it is called as a organelles. Okay, so simply meaning is these are the organs of cell, but we will not call them as organ, because cell itself is a part of organ, and therefore these are very small tiny things, so we call them as a organelles. Now organelles, people have classified it into again two ways. Some are having membrane bound, means they have their own distinctive membrane and therefore they are separate, and there are many which are not having such kind of organs. They are the cluster of molecules, they are coming together, but there is no uh, binding of a membrane, and therefore these structures are there, which are non-membrane bound. They are also given in our syllabus as a large assemblies of macromolecules, because these are the large assemblies. These are the molecules, and they are coming together to have a similar action. So we shall see how or which are the organelles which are membrane bound, and which are the organelles which are not membrane bound. So we have got like mitochondria, which are present in animal cells, plastid, etc. These are there in the plant or other cells. And non-membrane bound, there are many like ribosomes, spliceosomes, etc. Large protein complexes like proteasomes, DNA polymerases, holoenzymes, RNA polymerases, holoenzymes, then viral capsids, then the complex of GROL and GROS. We shall see all these membrane protein complexes, ATP synthesis, then large DNA and protein complexes like nucleosomes, centrioles, microtubule organizing center, then cytoskeleton, flagellum. Like that, many such things are there which are present, and we can go through these various organelles which are present, and how the organelles are there, and what are their brief functions. We can go for each organelle from this. That is, these are the structurally complex, and they are organized in part by interior compartments. They are enclosed by lipid membrane that resemble the cell membrane, so we call them as a membrane bound. Membrane bound means it doesn't mean that they are attached to the cell membrane, but they do have a similar membrane as the cell membrane is. Okay, but it may be slightly different than cell membrane, but they have got some membrane, and therefore they are demarked by a membrane. So if anything has to come in or go out from that organelle, it has to cross again a membrane. Okay, so these are there. Now the larger organelle like nucleus and vacuoles can be visible with the microscope, and they were the first biological discovery made after invention of microscope, that something has been seen. But later many techniques are evolved. There are two basic things, that is a microscopy that is called as electronic microscopy, which can give a very big what we can say increase in the uh, this size. And second thing is the fractionation. Fractionation means it is a centrifugal force which is applied, and because of the centrifugal force, the molecules, on the basis of their weight, they will be separated into it, and this separation will be giving the idea that is, these are the compounds with this kind of molecular weight, this molecular weight. And then after separation, there are many techniques which are there, which will allow to study these cells, and particularly these parts. So these are various ways by which this identification of what kind of agents are there is done. Now, let us see which are the eukaryotic organelles. We shall go one by one. As I told you, some are membrane bound, some are not membrane bound. Okay, membrane bound means they do have their own membrane. That is membrane bound. It doesn't mean, I repeat, that they are attached to the cell membrane. So, again, there are many organelles are present. Some cell may not have all the organelles. Some may have some organelles, some may have other organelles. Okay, because we have seen that cell is a structural and functional unit of the body or any organization, and in that some organs, some tissues may have some function, and therefore these may have specific organelles which are required, and some may not have all the organelles. So let us see what are the various organelles. First is cell membrane. We know basic function, that is, it separate the uh, cell interior portion of the cell from the exterior portion, the outer environment or extracellular fluid which is there, and it protects the cell from its external environment. We know about structure, it is a lipid bilayer, and this is a very typical one. We have seen a fluid mosaic model earlier uh, during absorption, how it is. So we'll not go for more details, but I'll suggest you this I am using from Wikipedia, because it is being constantly updated, and this is the field which is very rapidly growing. Many times the books may not have these details, and comparatively it is easy to go through. Particularly, may not be for all pharmacological topics, but particularly this cell biology and genetics, it is very rapidly evolving. Many people are working on it, and therefore this I feel it is more suitable than any book. Second benefit it is offering is, suppose now I am going for organelles, I want to go for detail of each organelle. So what I'll do is I'll click on the cell membrane. If I click, then I can have a separate topic of cell membrane. Can you see cell membrane? Okay, now all the details will be available from cell membrane. So if I am studying organelles, my everything, all organelles in details will be available for me. Okay, so I'll go in that way. See, this is all about cell membrane, and all this depth is covered, whatsoever is required for my knowledge or anybody's knowledge. So this much detail I am very sure is sufficient for you. Then if still you want to go ahead into it, then what are the integral proteins, what are various lipids, what are peripheral proteins, you have studied it earlier. Okay, and what are their importance. So these are, I'll click to the integral proteins. Okay, or I'll go for the peripheral proteins, and then I'll understand about this. So this is how it helps. All the functions are given. Already you have studied all these functions. So whenever I am going from for cell membrane, fluid mosaic model, I am repeating again and again. So this will help you to study in more depth. It will help you to understand the things in more details, what's required you can have, and from that it will give you all the things which are required as per our syllabus. I hope it's clear. So cell membrane is the first one. Then cell membrane causes, what is the function of cell membrane? Again, we'll go for brief detail of each, because we don't have that much time that we'll have liberty to go for each and every organelle in that much detail. But yes, we'll go for the gist very fast. After that, I'll just make a list and brief function first, and then we'll go at least 3 to 4 organelles a day. Like that, we'll try to finish. Uh, so cell membrane, then cell wall is typically present in plants and other things. It is made up of cellulose, and it is made up of peptidoglycans, rigid. It provides shape to the cell. It keeps the organelles inside the cell and does not let the cell burst due to changes in the osmotic pressure. And then next is chloroplast. Again, it is in the plant cell. It is doing the job of photosynthesis. It traps energy from sunlight. Now we are studying for all eukaryotes, which includes plants, animals, or all, particularly mostly multicellular one. Okay, chloroplast has got its own DNA, just like mitochondria we have. Okay, so it has got its own DNA. It is theorized to be engulfed by the ancestral eukaryotic cells. The word is endosymbiosis, means inside there is a symbiosis. Symbiosis means uh, staying with each other, helping to each other is called as a symbiosis. So it is a symbiosis. Endo means inside the cell. Then next is endoplasmic reticulum. You might have studied it earlier. Always it is, uh, or most of the time, it is present with the ribosomes. So we call it as a rough endoplasmic or smooth endoplasmic. So it is, the job is translation and folding of new proteins. If it is a rough endoplasmic reticulum, then expression of lipids. If it is a smooth endoplasmic reticulum, it is present in all eukaryotes. It is a single membrane compartment, but still it is a membrane compartment, means it is having a membrane. So rough endoplasmic reticulum is covered with ribosomes, so we call it as a rough. It has folds that are the flat sacs. Smooth endoplasmic reticulum has folds that are tubular in nature, and from that lipids are particularly uh, formed. Flagellum is for locomotion. It is present in some of the eukaryotes. Then next is Golgi apparatus. It is sorting, packaging, processing, and modification of protein. It is also a single membrane compartment. It is present in all eukaryotes. Then there are two basic units, cis and trans. That cis phase, it is the nearest to the rough endoplasmic reticulum. Trans phase farthest from the rough endoplasmic reticulum. So what happens is the uh, proteins which are formed from the uh, rough endoplasmic reticulum, they are taken up by this Golgi apparatus. They are sorted out. They are packaged. Some vesicularized. We have seen that, example, many neurotransmitters are put into vesicles. So they are vesicularized. They are put into vesicles. If some modification is required, like some cutting, pruning, something is required, then it is done, and then Golgi apparatus will able to put it into this. They will secrete as such or in a vesicles. Then mitochondria is well known. It is a unit for, or it is the organelle for energy production, particularly oxidation of glucose substance, and it causes the release of ATP as a energy molecule. It is a double membrane compartment. It is present in the most of the eukaryote. Why most? Because in plants we have seen chloroplast is present doing similar function. Constituting element of the chondriome, it has own DNA, and it is, theory is there that it has been engulfed by an ancestral eukaryotic cell. Again, endosymbiosis. It was actually separate portion, and then it is taken up by, and it is now functioning as a part of cell. Then nucleus, we know again about nucleus, and we have to see in more details about these all functions. DNA maintenance, it controls all the activities of cell. How it controls? By, it is a chemical, it doesn't have any other thing, but it do it by chemical signaling, chemical alterations. Okay, what's your function cell has to do? DNA is having all the codes and information of it. DNA will accordingly process itself accordingly. DNA to RNA transcription will be there, translation will be there, and accordingly, these proteins, lipids, whatsoever are there, these enzymes will be formed. and accordingly, because of the things which are produced, the cell will function of that particular time. Example, if we say that it is an endocrine cell, then this hormone will be produced because of the genetic structure or genetic codes which are available there. So thus it causes it is controlling the all activities of cell. It is a double membrane compartment present in all eukaryotes, and it contains bulk of genome. We know about genome, but still we may discuss it when we shall go for more or detailed studies in the next section of this. Then vacuoles are there. They are uh, responsible for storage, transportation. It helps to maintain homeostasis. These are the single membrane compartments. They are present in eukaryotes and they carry many functions. These are all major one, which are almost present in all, but there are some which may not be present in all, in which may be present in some particular cell or in only in some organs it may be present. Mitochondria and plastis, including chloroplasts, they have double membranes, their own DNA, and according to this endosymbiotic theory, they are believed to have originated from incompletely consumed or invading prokaryotic organism. So from that, these are coming. This is the belief. This is long history. Actually, we will not go into this. Then these are many other organelles which are present. Example is acrosomes, which is present typically on spermatozoa. It helps spermatozoa to fuse with ovum. It is a single membrane compartment. It is present in most animal only, not in plants. Then autophagosomes, they are the vesicles that sequesters cytoplasmic material and organelles for degradation. It is a double membrane compartment present in all eukaryotes. Centrioles are there. They are anchor for cytoskeleton. I hope if you, whenever you have studied this meiosis mitosis, you have seen the role of centrioles. So they are anchor for the cytoskeleton, organizes cell division by forming spindle fibers. Their structure is microtubule proteins. They are present in animal. They play a very important role in cell division. Then cilium is there. They are responsible for movement in or out of the external medium. So critical developmental signaling pathway these ciliums are having. They are also made up of microtubule proteins, and they are present in animals, protists, and few plants. Then next are uh, nidocysts. These are having the function of stinging, and they are typically present in these cindarians. Then eye spot apparatus, it detects light, it allows phototaxis to take place. It is present in green algae and other unicellular photosynthetic organisms like euglenids. Glycosomes, they are carrying out glycolysis. They are single membrane. They are present in protozoas like trypanosomes. See now, this if we remember that there are some typical organelles or some typical portions which are present in cell, just like we have seen ribosomes which are having 30s and 50s subunit in bacteria, and in human it is 60/40. So that gives an idea. That is how specifically the drugs which are acting against it can be developed. Then next is glyoxysomes. They are doing the job of conversion of fat into sugars. They are present into plants. Then hydrogenosomes, they are responsible for energy and hydrogen production. They are present only in few unicellular eukaryotes. Very few are there unicellular eukaryotes, and in that it is present. Lysosome is known. They are present in most of the animals. They are causing breakdown of large molecules like proteins, polysaccharides. Even they are functioning in various immune mechanisms because they are having hydrolyzing enzymes, and they can destroy the cells or microorganism viruses. Then melanosomes, these are the pigment storage. They are present in animals. So from that, melanocytes are coming. Mitosomes are there. They are playing a role in iron sulfur cluster, that is Fe and S assembly, and they are again present in some unicellular eukaryote that lack mitochondria, where mitochondria are not there and where these mitosomes are doing the job. Myofibrils are there, they are doing the myocyte contraction. They are the bundle filaments, they are particularly responsible for actin and myosin, suppose we have seen. They are present in animals. Then nucleolus is having pre-ribosome production. It is having action on protein, DNA, RNA. It is present in most of the eukaryotes. Then oceloid, it detects light and possibly shapes, allowing phototaxic to take place. And it is only present in the members of the family Varnoviaceae. Again, this is very typical, very lesser one. Then parentosomes, it is not characterized why it is present, what are the function, but it is present in fungi. Peroxisomes are again important in many places. If you remember earlier, some mode of action of peroxisome proliferating uh, this activating receptor PPAR, isn't it? You have seen them. So these are the peroxisomes. Breakdown of metabolic hydrogen peroxide. It is present in the all eukaryotes. It is a single membrane compartment. Then proteosomes are present in all eukaryotes, in most of the bacteria also. It is a very large protein complex, is a macromolecule proteins, as we said, and it is causing degradation of unneeded or damaged proteins by proteolysis. Then ribosomes, it is present in all eukaryotes as well as prokaryotes, but we have seen that there are differences, eukaryote and prokaryotes. They are having the job of translation of RNA into proteins, and that's they are made up of RNA proteins. They don't have any membrane. Okay, without membrane, they are, we can call them as a large macromolecular assemblies. In our syllabus, this word is given, large macro organelles, and then next to that is large macromolecular assemblies. So these are the large macromolecular assemblies. Then it is present in all eukaryotic cells. Then stress granules, that is it is doing the job of storage of messenger RNA. It is a membraneless messenger RNA, that is RNA protein complexes. It is present in most eukaryotes. Then tiger domain, these are the mRNA encoding proteins. They don't have membrane. They are present in the most of the organisms. Then vesicles are there. They are responsible for material transmitter transport. We have seen various vesicles. If you remember, last year we have studied about razor pin blocking vesicular transport. So these are the vesicles. They are single membrane compartment. They are present in all eukaryotes. And other important structures are cytosol, endomembrane system, nucleosomes, microtubules, cell membrane. Okay, so these are all variety of structure which are present, and these all structures are playing important role in the cell physiology. So each organelle, whatsoever is present in a cell, plays very very important role in the functionality of the cell. Now, similarly, we shall have some brief idea about prokaryotic. As such, you might have studied all this in microbiology last year, but let us have a fast look about prokaryotic, because latter will concentrate more and more on eukaryote, particularly on human and animal related cells, and will give comparatively less this one less importance or less time for prokaryotic organelles. Whatsoever basics are there important only, we'll try to keep in mind. We'll not go in more details about this. So these are the prokaryotics. As these are unicellular, and they do have very little functional activities. So they are not structurally as complex as eukaryotes, because eukaryotes have multicellular, rather multi-tissue, multi-organ. These organisms are there, and therefore they have very complex and multiple functions they have to do. So for that, eukaryotes have got more varieties of cell. more varieties, more structural and functional varieties, and because of that, accordingly, more and more organelles and more type of all these system which is kept, or physiological mechanisms are made. And they were once thought that they don't have any internal structure, which are enclosed by lipid membranes, and they were often viewed as a little internal organization, and they lack cellular compartments. But then, later, as the study went on, it was found that yes, they do have various things. You can see these figures which are there, figure A and B. So, see, I was telling you about this electron micrograph. So, these are the developments which happen, because earlier microscope was not able to do up to nucleus only, and it will able to show the this one. But then after arrival of these various technologies, that is particularly like electron microscopy, which has got very high, what we can say, the capacity to increase the size. So this allows highlight some carboxysomes. Then the images are there of carboxysomes which are there, and see the size is 100 nanometer. So these are of nano level structures are there. So what is actually emphasis is that because of the invention of various technology, and technology are still coming out, and because of all these technologies, more and more idea, more and more knowledge about various cells and their structures, their functions are there. Because this all techniques simultaneously, many techniques, we shall discuss these various techniques also sometime. And but this, if you remember that because of all this technique, the science is growing like anything. Those who are interested, they can search about human genome project, and it was in early uh, of this century when 2000 started. During that, the project was undergoing, and that has related to formation, this searching about human genome, that how many genes are there, for what they are coding. And it was found that we don't know many thing about because genes are there which are capable to produce many kind of proteins, but we are not aware about their functions. So, one by one, slowly, because of all these techniques, all these advances, we'll able to understand, and therefore, the new techniques, new kind of drugs are arriving. You might have seen the increase in, just now we have seen recently in immunology. Earlier, very few things were available. Even immunosuppressant was there, hardly it was maximum 4 5 drugs were there. Now there is a, we have got a specific uh, immunosuppressant agents for various this one, various kind of immunological disorders or autoimmune disorders. And why this is, because we have understood what kind of activities are going on, because this more and more studies about cell, that resulted into more and more information. So it was understood that these are some antibodies are there, autoimmune structure are there, these antibodies are doing. So can we block these antibodies with something? So we have got antibodies against these antibodies, or antibodies against this protein, and because of this we could have various kind of monoclonal antibodies or variety of such proteins. So maybe in future, most of the things will be going in that direction, wherein the pharmacological drugs which are having actions at multiple places, and because of multiple places, more chances of adverse effect, they will be slowly come down. So we were discussing about these prokaryotic organelles, and initially it was thought that they don't have many things. It is just simple cell which is present, but later again it was found that there are many organelles are present, and these compartment are also present, and later these compartments are checked, and what are various organelles are present. So these are variety of organelles they are present. Example of these are uh, anamoxosomes. These are responsible for anaerobic ammonium oxidation. and they are present in particular some of the typical species of bacteria like candidatus bacteria within Plactanomycetes families. Then carboxysomes are there. They are doing the job of carbon fixation. Uh, the, the structure wise, they are either membrane structure or not membrane structure. So it's not difficult. They are present in some of the bacteria. Then chlorosomes are present. They are doing the job of photosynthesis. These are macromolecular complex is present. It is present in the green sulfur bacteria. Flagellum is present in most of the bacteria, those which are having motility. As I told you yesterday, you might have seen it in the experiment of motility that microbiology. Then magnetosomes are there. They are having magnetic orientations, and they are present in uh, the bacteria. That is magnetotactic bacteria are there. That families are there. In that it is present. Then next is nucleoids are there. Their job is to do the maintenance of DNA, transcription to RNA. They are part of the DNA protein and they are present in the prokaryotes. So, pilus is another type of organelle. It is uh, doing the adhesion to the other cells for conjugation or to a solid substrate to create a motile forces. It is a hair-like appendage that is sticking outside, which is not flagella. It is different, and it is present in the prokaryotic. It is sort of doing the job of anchoring, what we call it as a plasmid is very important. Plasmid is the structure which is appendages to DNA. You might have studied this plasmid last year in microbiology. I'm very sure. If not, just revise it sometime because we may use this somewhere. We may use this information at some place. These are responsible for DNA exchange. You might have seen all the process of conjugation, etc. They are part of the circular DNA and they are present in some of the bacteria. Then ribosome, we have seen the differences. In human being, it is 80s, or in animals, it is 80s, it is 70s. In 70, it is 50-30. In human, it is 60-40. Uh, when they come together, it becomes 70s, therefore, and when they are separate, the total becomes higher. So here, if they are separate, 50-30 is 80. But when they are coming together all over, then it is all both subunit when come together it becomes 70s. Similarly in human being it is 80s, but when they are separated it is 60-40. The job is translation of RNA into proteins. It is present into bacteria and such cells. Then next are thylakoid membranes. They are responsible for photosynthesis and they are mostly present in the cyanobacteria which are doing the photosynthesis. Apart from that, there are many other hypotheses are there, many other structures are there, or biogenesis of how organelles are biogenesis, how membrane vesicle trafficking taking place. These are part of an endosymbiotic theory, as I told, but these are not of our importance, so we'll not go for this now. We shall discuss various organelle one by one uh, from next class, and if you have read it well before going, then we can do it faster because we cannot put more and more time into this. So please go through this. Basics already are there, and just we'll add up into that. The most important thing during this whole process, why we are studying, we have to understand, because if we understand cell and their function, then we'll understand the pathology. Okay, and if we understand why it is there, then what can be the corrective measures that can be easy. Okay, therefore we are going for all these processes, and at many places how this, because earlier it was not very clear. It was logically found out what are the pathologies or what are the reasons, but nowadays we are going for almost even minute level, more than molecular level or genetic level for all these diseases. And at many places now we are able to understand that what molecular or what genetic problems are there for which are responsible for diseases. So maybe in future, whole concept of treatment will shift from drug-based pharmacology to the biotechnology-based pharmacology, and therefore we are studying all this. Therefore, we are trying to understand what will be the nature, what are the things which are present in the cell. And as I told you, with the evolution, that is growth of the technology, because it is not just single simple biology actually. So many sciences are involved into that. So again, before going for organelles, we'll try to have a one look of it, so that you will understand that how the science is growing, or how it is a multidisciplinary science. And because of this multidisciplinary activities, the ability to understand more about cell is possible. Once we understand more detail about each and every cell type, their functions, what component are responsible for functions, then it is becoming easier to pinpoint their role in various diseases, or in various physiologies and pathologies, and then it becomes easier to try to solve it at that level. We were earlier doing at cellular level most of the time with pharmacological measures. Now, can we do it at that level? That is happening. Actually, the things are going in that direction. So, I am very sure this much is clear to you. Try to go for the details which you have studied earlier. If questions are there, we'll put it on WhatsApp group. Then someday we'll keep the session for difficulty solving. Okay. Fortunately, this is the good one, good source of information than books, because this is very very latest. If you see the latest edition, sometime it will be in 2000. People have done it. Okay, so that will help you to understand latest things. But we'll try to restrict what's there up to our course, and will not go into more details. Okay, so take care. Uh, Use your time in studies, read the basic things till other classes are not there, and definitely it will help you a lot to make the basics more better. So take care and have a good time.